Namaste to all students. Myself Jeevan Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about microscopy session, and this session is related to cell biology course of program BTech FBT and BSc FST second sem. So before starting this session, you have to know about the definitions of microscopy. So the microscopy is the basic tool of cell biology, and recent advances in imaging technology have allowed an unprecedented amount of information to be collected from microscopic analysis. There are various types of the microscopic techniques which are used include bright field microscopy. This is traditional microscopy where cells are illuminated by visible light. Bright field microscopy gives a general picture of cell function. Although that information is not very detailed or specific, as animal cells lack cell walls, bright field microscopy may use special techniques such as phase contrast to show cellular structures in more detail. Bright field microscopy allows imaging of live or fixed, means dead cells or tissue. Second type is electron microscopy. This microscopy uses a focused beam of electrons instead of light. Electron microscopy permits a much higher magnification of specimens than light microscopy, and it is useful in obtaining detailed information about subcellular structures. Electron microscopy requires extensive progressing and so can only be performed on fixed specimens. Transmission electron microscopy provides a cross section of a specimen, while scanning electron microscopy gives a three-dimensional image of the surface of a specimen. Third type is fluorescence microscopy. This kind of a microscopy basically uses fluorescent material to indicate the structure in a specimen. Fluorescence occurs when light of one wavelength excites a material and causes it to emit light of a different wavelength. Most fluorescent material give off visible light after excitation by ultraviolet light, and the structures may be naturally fluorescent, which is also known as autofluorescence, or they may be labeled with a compound which is fluorescent. For example, DAPI, it is a dye which binds only to the DNA material, and the DNA nuclei of the cell stained with the DAPI emits a blue light under ultraviolet light. Next type of a microscopy is time-lapse microscopy. Many cellular processes, for example, mitosis, occur over a period of time which is not practical for direct observation. Imaging cells over a period of time, for example, photograph is taken every 20 minutes for 20 hours, allow us to combine these images in a movie, which compresses a long time period into a shorter one. So in this session, now come to the very famous microscope working principle and its procedure. So it is compound microscope. So first of all, before understanding the principle and the procedure of compound microscope, we have to know about the definition of the compound microscope. So compound microscope is an indispensable instrument in any biological laboratory. It is used for passive observation of a structural detail of a cell, tissue, or organ in the sections. A modern compound microscope has following structural components. It is having non-optical components, which includes base, which is foot, pillar, arm, which is limb, and inclination joints, stage, and body tubes. On the other hand, and draw tube, rack and pinions, adjacent screws, automatic stop, and these all components come into the non-optical region. And if you are talking about the optical components of compound microscope, so it includes diaphragm, condenser and reflector, which is also known as mirror, along with the objective lenses and ocular lens or eyepiece. So compound microscope structurally can be divided into two sections, non-optical component and optical component. Now come to the detailed observation of non-optical component. So first one is the base, which is foot. It is U or horseshoe shaped metallic structure that supports the whole microscope. Second component is pillar. It is a short upright part that connects to the base as well as arm. Third one is the arm, which is also known as limb. It is a curved metallic handle that connects with the arm by inclination joints. It supports stage and body tube. Fourth one is the inclination joint. It is used for tilting the microscope if required for observation in sitting position. Fifth one is the stage. It is a metallic platform with a central hole fitted to the lower part of the arm. Microscopic slides held on the stage by either simple side clips or by a mechanical stage clip. Fourth component is body tube. It is meant for holding ocular and objective lenses at its two ends. The end holding ocular lens is called head, while the end containing 3 to 4 objective lens is called nosepiece. The body tube has an internal pathway for the passage of light rays which form the enlarged image or microscopic objects. Seventh component is draw tube. It is a small tube that remains fixed at the upper end of the body tube and it holds eyepiece or ocular lens. Eighth component is rack and pinions. The microscope has a rack and pinion attached either to the body tube or the stage for bringing the object under the focus. Ninth component is the adjustment screws. So these are two pairs of the screw for moving the body tube in relation to the stage, larger for coarse adjustment and smaller for the fine adjustment. In fine adjustment, the body tube or stages move for extremely short distance. In coarse adjustment, the body tube or stage can move up and distance can be large. So in coarse adjustment is meant for briefly objective lens at a proper distance from the object so as to form image of the same at the ocular end. And fine adjustment is required to obtain sharp image. 10th component is automatic stop. So it is a small screw fitted at the lower end or rack on the rack and pinion. It is meant for stopping the downward sliding of the body tube so as to prevent the damage of the objective lens at the slide. So these are the components related to the related to your non-optical region. Next one is the optical component. In optical component, first component is diaphragm. It is fitted just below the stage for regulating the amount of light falling on the object. Diaphragm is of two types. Disc diaphragm can be and iris diaphragm can be adjusted in compound microscope. Second component in optical is condenser. It is attached below the diaphragm. Condenser can be moved up and down to focus the light on the object. Third material is reflector, which is also known as mirror. It is attached just above the base. Both its surface bear mirror, plane on one side and concave on the other side. Plane side is used in strong light and concave side is used for weak light. Reflector directs the light on the object through the condenser and diaphragm system because it is just below under the diaphragm and condenser. So that's why it provides the light to the object passing by the condenser and the diaphragm system. Fourth one material is objective lenses. So they are fitted over the nose piece. Objective lenses are of two or three types: low power, which is commonly 10x or 5x, high power, commonly 45x, and oil immersion, commonly 100x can be more. An objective lens is not a simple lens but compound lenses. It forms real inverted image of the object inside the body tube. Fifth component is ocular lens or eyepiece. It is lens through which image of the microscopic object can be observed. It also takes part in the magnification depending upon the magnification. The eyepiece is of four types: 5x, 10x, 15x, and 20x. Advanced microscope has two eyepieces so that both the eyes can be used. Microscopic head have device for using two eyepieces is called binocular head. It contains a number of internal mirrors and prism for the passage of the light. These are the components of compound microscope, and compound microscope, as I have already told you, divided into two sections: non-optical region and optical region. And in our previous slides we have discussed about the components of these two sections. Here we are going to understand detailed structure of compound microscope through this diagram. In this diagram you can simply see eyepiece. This is the draw tube, this one is the body tube, and this large one is the coarse adjustment, and small piece is for fine adjustment. This is nose piece, this is automatic stop to just prevent the damage, and this one is the arm, and this small joint is inclination joint. This is foot. These all are come into the non-optical region. Now come to the optical components. So these are the eyepiece will come into the in the objective region, in the optical region, and this one is the objective also come. Diaphragm, condenser, concave, mirror, light reflector, which is also known. So these all come into the optical region, but stage, slide, clip, these all come into the non-optical region. So this is the structure of compound microscope. Now come to the working principle of the compound microscope. So the compound microscope is most commonly used in clinical and educational laboratories. It has a combination of the lenses that enhances both magnifying power as well as the resolving power. It has a combination of the lenses that enhance both magnifying power as well as the resolving power. The specimen or object to be examined is usually mounted on a transparent glass slide and positioned on the specimen stage between the condenser lens and the objective lens. A beam of light from the base is focused by a condenser lens onto the specimen. The objective lens picks up the light transmitted by the specimen and create a magnified image of the specimen called primary image inside the body tube. This image is again magnified by the ocular lens or by the eyepiece. So this is the sum up of this session, and this session is powered by digital version 2.0 Jayoti Vidyapeeth Women's University Jaipur. If you you have any query, then mention in the comment box. I will resolve it. Thank you.